Dear students, welcome to our science episodes, and let's discuss together today part 2 of cell division. In the previous session, we discussed cell division, especially mitotic cell division or mitosis. Today, we are going to discuss meiosis. In this picture, it's written that meiosis plus fertilization equals sexual reproduction. This means that meiosis is related to sexual reproduction, where two different gametes are needed from two different parents, and they will join together, they will combine together in a step called fertilization, and a new organism which is genetically different from the parents will be produced. Meiosis is separated into two stages, meiosis 1 and meiosis 2. Cells in the testis, in the male body, or in the ovary belonging to the female body divide. They divide to produce sex cells or reproductive cells or gametes. These gametes are either sperms or ova. In flowering plants, meiosis takes place in the anther, which is the male organ in the flower, to produce pollens. These pollens contain sperm cells. And in the pistil, or in the flower's ovary, to produce the ova. Meiotic cell division produces 4 cells instead of 2 cells, with half the number of chromosomes of the parent cell. Also, we have here a preparation phase. It's called an interphase, where the cell will prepare itself for division by copying or doubling the genetic material. Meiosis comes in 2 stages of cell division, meiosis 1 and meiosis 2. Let's discuss meiosis 1. In prophase 1, the chromosomes will condense and the pairs will arrange together. Each pair consists of 4 chromatids called tetrad, and the nuclear membrane disappears. Every 2 homologous chromosomes start to move away from each other. And each chromosome consists of 2 chromatids connected by the centromere. So homologous means identical. So identical chromosomes begin to condense together. Spindle fibers start to appear and they become attached, the chromosomes become attached to the fibers, and crossing over occurs. Can you notice that the changes in the prophase 1 are many? Yes, this is the longest phase, and this what characterized or make meiosis 1 special, prophase 1. In metaphase 1, and as usual, the chromosomes will arrange in the equator of the cell. Pairs of chromosomes will arrange in the equator of the cell. In anaphase 1, every 2 homologous chromosomes move away from each other as the spindle fibers shrink, and chromosomes move toward the opposite poles of the cell. In telophase 1, the nuclear membrane appears again, and the 2 cells will separate, each contain half the number of chromosomes of the mother cell. If you noticed in meiosis 1, there are special changes. The prophase has many changes, and the end result is 2 cells with half the number of chromosomes. While meiosis 2, each cell resulted from meiosis 1, each cell will undergo meiosis 2. That's why the end result is 4 cells. And meiosis II is typically or identical to mitosis. So if you know mitosis well, or you can remember the steps of mitosis, it's the same as meiosis II. It aims just to increase the number of the produced cells, gametes, and the division is a way similar to mitotic division. I told you, if you can remember the steps of mitotic division or mitosis, it's the same as meiosis II. At the end of the telophase, 4 cells are produced, and each contains half the number of chromosomes of the parent cell. The crossing over phenomenon. The crossing over phenomenon takes place in prophase 1. So in meiosis I, in the prophase stage. At the end of this stage, some pieces of the 2 inner chromatids are exchanged together. Why? Why chromosomes are exchanging parts? To produce new genetic arrangements. This is called crossing over. And that's why you are not identical and similar to your sibling, to your sister or your brother, because of this crossing over phenomena. That's the importance of crossing over. It results in gene exchange. This is the importance of crossing over. It results in genes exchange between the chromosomes. So, it's an important factor or an important cause for the variation of genetic traits among the individuals of the same species. Let's talk about cancer. It's when the body cells divide without control. The mass resulted from this division, we call it, or the doctors call it, tumor. How could it be detected? Let me tell you that the Egyptian scientist, Dr. Mustafa Said, discovered a way to detect the cancer cells. And this technique includes loading proteins, having the ability to attach to the cancerous cell secretions with nanomolecules of gold, and then injecting them into the patient. So we will get nanomolecules of gold and combine them with proteins which can attach to the cancerous cell to detect the cancerous cell and monitor the disease through a microscope. What about its treatment? Also using the nanotechnology, scientists developed smart microscopic bombs. And these microscopic bombs could penetrate the cancer cell and kill them. They were used to kill the cancerous cells in experimental mice. And the mice suffered from cancer were able to live 300 days after this treatment. As for the mice that didn't receive the treatment, they didn't live more than 43 days. The treatment is by focusing laser with a certain degree to gold molecules. And these molecules will absorb the light and convert it into heat, which will burn the tumor cells when it will stuck to the gold molecules. Now, let's see this Venn diagram to compare and contrast mitosis and meiosis. What are the similarities and what are the differences between mitosis and meiosis? Mitosis is related to the asexual reproduction, while meiosis is related to sexual reproduction, where two parents and gametes have to be there. Mitosis' aim is the growth of living organisms and the repair of the damaged tissue, while meiosis is mainly for production of gametes. Somatic cells divide by mitosis, except the nerve cells and the red blood cells, while reproductive cells divide by meiosis. Mitosis produces 2 cells, while meiosis produces 4 cells. The 2 cells produced as a result of mitosis are diploid, having the complete set of chromosomes, while the 4 cells produced from meiosis are haploid, having half the number of the chromosomes. Mitosis is 1-stage cell division, while meiosis are 2 stages, meiosis 1 and meiosis 2. And of course, meiosis has the crossing over phenomenon we discussed before. Both of them are cell division. Both of them produce new cells. Both of them start with diploid parent cell and preceded by interphase. Now, let's fill in the blanks together. Meiosis occurs in flowering plants in the anther to produce what? Pollens. Number 2, crossing over phenomenon takes place in which stage of which cell division? Think about it. Which is more special, meiosis 1 or meiosis 2? It's meiosis 1. Why? Because the prophase of meiosis 1 is so special, having many changes. Therefore, the answer is prophase stage of meiotic cell division. Number 3, if the number of chromosomes in a liver cell is 46, then the number of chromosomes in an ovum is, and please remember that the ovum is a reproductive cell, a sex cell, a gamete, so it contains half the number of chromosomes of any somatic cell. Now let's watch this video about meiosis.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis I. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving two pairs of sister chromatids. Chromosomal material is exchanged between the two pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no two siblings, aside from twins, are genetically identical. There are several more key steps in prophase I. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis I is called metaphase I. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase I. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis I are telophase I and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis II. Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase II, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis II began with two cells, and each of those cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are gametes. Two gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Dear students, thank you for watching and see you later in other science episodes.